picture this with me for a second. Imagine a perfectly normal, delicious donut.
Okay, I'm with you.
Right. So now imagine you have a straw passing right through the hole in the center of that donut. And give that donut a few years, maybe a few decades, and it slowly starts to swell. The dough expands inward, get, puffier and puffier, squeezing that straw so tightly that eventually, well, absolutely nothing can get through.
Which is, frankly, a terrifying thought.
Exactly. And for about 50 to 60% of men in their 60s and up to a staggering 90% of men in their 70s, that donut is their prostate gland. And that straw is their urethra.
Yeah, we were talking about benign prostatic hyperplasia, or BPH.
Right. And it's basically...
It's the absolute definition of a plumbing nightmare. I mean, it drastically affects a patient's daily existence.
Oh, totally.
We're talking about hesitancy, weak flow, incomplete emptying. It's chronic. It's exhausting. And for decades, the medical community has treated it more like a mechanical failure than a complex biological mystery.
Well, our mission for today's deep dive is to unravel that exact mystery. We're pulling from this really groundbreaking, newly published 2026 study in the journal Cell, Death, and Disease.
Yeah, it's a fantastic paper.
It really is.
Right.
And we're going to unpack a hidden, epigenetic switch that causes this exact prostate enlargement. And even better, we're going to look at a potential new drug that could, flip that switch right off and stop the swelling in its tracks.
Which is huge.
But before we get to the cure, let's establish the baseline here. What is the current standard of care for a patient who walks into a clinic today with this issue?
Right. So the current standard relies heavily on, primarily two classes of drugs. You have your 5-alpha reductase inhibitors, which basically attempt to shrink the prostate by blocking specific hormonal pathways. And then you have alpha-adrenergic receptor antagonists.
That's a mouthful.
It is, yeah. But they essentially try to just relax the muscle fibers around the donut, to borrow your analogy, just to let fluid pass through.
So we're basically just trying to chemically pry the donut hole open.
That is the goal, yeah. But they often fall short. I mean, these drugs manage symptoms to a degree, but they frequently fail to stop the underlying progression of the disease itself.
Right.
Roughly 30% of these patients eventually end up on an operating table, requiring surgery to literally, physically core out the center of that overgrown prostate.
Oh, wow. That sounds intense.
It is. And the fundamental problem here is that the exact etiology, the root cause of why this tissue starts growing out of control in the first place, has just remained elusive.
But we've known for a long time that inflammation is involved, right?
Oh, absolutely. Chronic inflammation is a major player. I mean, we see heavy inflammation in over 70% of men with BPH. But the missing link has always been understanding how a swollen, angry environment actually, forces the tissue to physically multiply.
Right. And let me push on that for a second, because if we know inflammation is involved, why is it such a mystery? You get a cut on your arm, it gets inflamed, it swells up. Why wouldn't the prostate just be doing the exact same thing?
Well, because the swelling of a cut on your arm is mostly just fluid rushing to the area.
Okay.
But in BPH, the prostate isn't just full of fluid. The actual cells are multiplying.
Ah, gotcha.
Yeah, the tissue is getting denser and larger because the cells are dividing completely out of control. So inflammation is the spark, sure, but the researchers needed to find out how that spark was communicating with the DNA of the prostate cells to, explicitly tell them to multiply.
Which brings us to the methodology of this study. And to find that spark, the researchers didn't just look at the tissue as a massive block, right? They zoomed it all the way in.
They did. They used single-cell RNA sequencing alongside traditional bulk RNA sequencing.
And the distinction between those two techniques is basically entirely why they found the mechanism. Let's use the smoothie analogy for this, because I think it really clarifies why this is such a breakthrough.
Oh, I love the smoothie analogy. Go for it.
Okay, so bulk RNA sequencing is like taking an entire mixed fruit smoothie and trying to guess the exact ingredients just by tasting it. There are strawberries and bananas in there somewhere, but it's all blended together.
Right.
But single-cell RNA sequencing is like magically taking that smoothie apart and analyzing every single microscopic piece of fruit individually. It tells you exactly what individual specific cells are doing at any given moment.
Exactly. It's incredibly precise. And when they compared the individual cells of human BPH tissues to normal, healthy prostate tissues using this method, the data was just incredibly clear.
What do they find?
Well, first, the BPH tissues had massive inflammation skulls, which we expected. But more importantly, they noticed a very specific gene acting up across multiple cell types.
Or like which ones?
We're talking fibroblasts, basal cells, luminal epithelial cells, and this specific gene was churning out massive amounts of a protein called JMJD3.
Wait, JMJD3, also known as KDM6B in some literature, right?
Yes, exactly. So they find this JMJD3 protein surging in the diseased tissue.
Okay, but finding it at the scene of the crime doesn't necessarily mean it committed the murder, right? Like, how did they prove that the inflammation actually caused the JMJD3 to surge?
That's the million-dollar question. So they moved from human samples to a tightly controlled laboratory environment. They took normal human prostate cell lines, specifically structural cells called WPMY1 and lining cells called RWPE1.
Okay.
And they introduced a substance called LPS lipopolysaccharide.
Oh, right. LPS is it's a toxic component found in the outer membrane of certain bacteria, right?
Exactly. It's an inflammatory toxin. When you drop LPS onto living tissue in a petri dish, it triggers this massive artificial inflammatory response.
So you're basically simulating the angry, inflamed environment of an aging prostate in a dish.
Spot on. So they blast these normal, healthy cells with this LPS toxin, and what happens? The levels of JMJD3 just absolutely skyrocketed.
Oh.
The inflammation directly forced the cells to produce enormous amounts of this protein. They established a clear, direct causality.
Okay, so just to pause and make sure I'm tracking the biological chain of events here, and let's do a quick recap for you listening.
Right.
We have the aging prostate, which for various reasons develops chronic inflammation.
Right.
That inflammatory environment acts like a biochemical spark, and that spark forces the local prostate cells to flood themselves with this specific protein, JMJD3.
Exactly.
But that still leaves a pretty massive gap, which leads to our first big question for you to ponder. If your cells are suddenly flooded with JMJD3, what exactly is it physically doing to your DNA to make those cells multiply?
And to answer that, we have to take a step into the world of epigenetics.
This is where the mechanics get genuinely fascinating. So think of your DNA as a massive, like 20,000-page instruction manual for building and operating your body. Every single cell in your body contains the exact same manual.
Which is pretty wild when you think about it.
It is. But epigenetics are like little sticky notes placed on the pages of that manual that say, read this page constantly or skip this page entirely. You aren't changing the text of the manual itself. You're just changing how the cell reads it.
Right. And that is exactly how a prostate cell knows to act like a prostate cell and not, say, an eyeball cell.
Right. So the body uses chemical tags as these sticky notes.
Yes, and one of the most important inhibitory tags, a tag that acts as a skip this page sticky note, is a specific histone modification called H3K27E3.
Okay, H3K27E3. We don't need to get bogged down in the chemistry of the acronym, but fundamentally, this tag acts as a molecular do not disturb sign.
Exactly.
Right?
It physically blocks the cellular machinery from reading whatever gene it's attached to. It keeps genes completely silenced.
And in a healthy adult prostate, you desperately want certain growth genes silenced, right? So the gland doesn't just keep growing indefinitely.
You absolutely want those do not disturb tags firmly in place on growth genes. But, and here is the critical plot twist of the study.
Okay, let's unpack this.
That JMJD3 protein, the one that surged during the inflammation, it is categorized biologically as a histone demethylase.
Meaning?
It's an epigenetic eraser. Its entire biological function is to find those specific H3K27E3 do not disturb tags and literally rip them off the DNA.
Wait, really? So the inflammation produces an eraser. But does this eraser just wipe out everything? Because ripping all the sticky notes off the DNA everywhere sounds like it would just result in pure chaos, like destroying the entire cell.
That was the exact question the researchers had. To test the sheer power of this eraser, they genetically engineered those normal prostate cells we talked about to artificially overexpress JMJD3.
So they flooded the cells with the eraser without even needing the inflammation spark.
Exactly, just to see what would happen if the eraser ran wild, all on its own.
I'm guessing the cells kind of lost their minds.
Completely. The researchers observed a massive acceleration in the cell cycle. See, normal cells spend most of their time sitting quietly in a resting phase, the G0, G1 phase.
Right.
But with the sudden influx of JMJD3, these cells were rapidly pushed out of that resting phase. They rushed directly into the active S and G2M phases, basically copying their DNA and physically dividing.
Wow.
They began to proliferate uncontrollably, mimicking the exact overgrowth we see in human BPH.
The eraser removed the brakes, and the car just floored it. So let's synthesize this before we look at the exact target. We know epigenetics are the sticky notes on our DNA. We know there's a specific do not disturb sticky note, H3K27NE3, that stops cells from dividing.
Right.
And we now know the inflammation-triggered protein, JMJD3, acts as an eraser that rips off those sticky notes, causing the prostate cells to multiply.
Perfectly summarized.
Which begs the question for you listening. But what specific monster of a gene is being unleashed when that stop sign is removed?
And this is key, because JMJD3 doesn't just randomly erase tags everywhere, it actually has a highly preferred target.
And how did they find it?
To locate that target, the researchers used a molecular tracking tool called ChIP-seq.
ChIP-seq. That functions essentially as a molecular GPS tracker, right?
Yeah.
Telling you exactly where on the giant DNA map a specific protein is hanging out.
Exactly. They effectively dropped a pin on the exact gene the eraser was targeting. And when they combined that GPS data with RNA sequencing, which tells us which genes are actively being printed into proteins, a very specific pathway lit up.
Which one?
The TGF-1 signaling pathway.
Oh, wow. Transforming growth factor, beta-1. I mean, for anyone who studies cellular biology, that name immediately sets off alarm bells.
It really does. It is a heavy hitter cytokine. Under normal circumstances, it helps with localized tissue repair and things like that. But the ChIP-seq data revealed something incredible. The do not disturb tags are normally heavily clustered right on the promoter region of the TGF-1 gene.
Okay, the promoter region, that's basically the ignition switch of the gene. So normally the ignition is locked down tight and the key is thrown away.
Exactly. But if we connect this to the bigger picture, JMJD3 specifically targets that exact ignition switch.
Yeah.
It binds to the promoter of TGF-1, erases the do not disturb tags, and turns the TGF-1 gene on permanently.
It hotwires the bulldozer.
That's a great way to put it. And once TGF-1 is unleashed inside these prostate cells, the study shows it does two deeply problematic things.
Right, let's break those down.
First, it promotes rapid cell growth by ramping up the cell cycle proteins. We're talking massive increases in proteins like CDK2, CDK4, and cyclin D1.
So if the cell cycle is a ticking clock, TGF-1 basically adds a massive motor to the gears, making them spin faster and faster.
Exactly. And second, and maybe more importantly, TGF-1 actively stops apoptosis.
And apoptosis is programmed cell death, right? Normal cells have a built-in self-destruct timer to ensure they don't overpopulate or outlive their usefulness.
Right, it's a crucial safety mechanism. So TGF-1 effectively cuts the brake lines on cell death. You now have an environment where prostate cells are multiplying at breakneck speed, and they are outright refusing to die.
So the accumulation of all those immortal multiplying cells is literally the physical dough of the donut swelling up and crushing the urethra.
That's exactly what's happening mechanically, yes.
We now have the complete map of the enemy's battle plan. Let's recap this before the final phase. The eraser, JMJD3, removes the stop sign specifically on the TGF-1 gene, flooding the prostate with growth signals and stopping cell death.
Right.
So the question to think about now that we know the exact mechanism, how do we throw a wrench in the gears and stop it?
And here's where it gets really interesting. This brings us to the therapeutic breakthrough of the paper, a small molecule inhibitor named GSKJ4.
GSKJ4, what does it do?
This molecule was specifically designed to bind to and block the activity of JMJD3.
Oh, nice. So it's essentially handcuffs for the epigenetic eraser?
Yes, exactly. The researchers first tested these handcuffs in the Petri dish using a cell line called BPH1.
And those are cells derived directly from actual human benign prostatic hyperplasia tissue, right?
Correct. These are the real deal hyperactive swollen prostate cells.
So they dropped this GSKJ4 drug onto them. What actually happens to the cells?
The reversal was incredibly effective. When the BPH1 cells were treated with GSKJ4, the researchers observed immediate cell cycle arrest. The rampant division just stopped.
Wow.
Furthermore, it induced apoptosis. It turned that self-destruct timer back on, allowing the body to start clearing out the excess cellular buildup.
The study also noted it lowered ROS, right? Reactive oxygen species.
Yes, it did.
And just to clarify for everyone, think of ROS as the toxic cellular exhaust fumes that build up when a cell is stressed, inflamed, and overworked. So the drug lowered those exhaust fumes, basically calming the entire environment down.
Exactly. By blocking the eraser, GSKJ4 allowed the cell to naturally put its do not disturb tags back onto the DNA.
But, fixing cells in a petri dish is one thing. The real test of any potential therapeutic is whether it actually works inside a living, breathing biological system.
Oh, absolutely. And to prove this wasn't just an in vitro fluke, the researchers set up 2 distinct animal models.
Let's walk through these because I think this is the bridge between a neat lab trick and a future human therapy.
Yeah.
Model number 1 was the rat LPS model.
Right. So they took rats and injected that bacterial LPS toxin we talked about earlier directly into their prostates to trigger the massive chemical inflammation.
Okay.
And as expected, the rats developed severely enlarged prostates. Their prostate index, which is basically the weight of the prostate relative to their total body weight, just shot up.
Makes sense. But then they gave a group of these inflamed rats the GSKJ4 drug over a 4-week period, right?
He did. And the prostate enlargement was significantly suppressed.
That's amazing.
Yeah, and to prove why it was suppressed, they looked at the tissue under a microscope and stained it for a specific biomarker called Ki-67.
Oh, Ki-67 is a fascinating tool in pathology. It's a protein that is strictly associated with cell proliferation. Like, if a cell is resting normally, Ki-67 is totally undetectable.
Right.
But if a cell is actively dividing, Ki-67 lights up. It acts like a glowing neon open for business sign for cell division.
Exactly. And in the untreated inflamed rats, the prostate tissue was just glowing with those Ki-67 neon signs. The cells were dividing everywhere. But in the rats treated with GSKJ4,
What is it?
the Ki-67 levels plummeted. The drug successfully halted the pathological cell division in a living animal.
Okay, so that covers a chemical trigger. But we know human aging is, well, complicated, and BPH is heavily tied to hormonal changes as men get older.
Right.
Which brings us to model number 2, the testosterone mouse model.
Yeah, they gave mice daily doses of testosterone, which predictably induced BPH and severe tissue inflammation. This mimics the hormonal driver of the disease rather than just a raw bacterial chemical response.
So they took a group of these hormonally overloaded mice and co-treated them with our eraser-blocking drug, GSKJ4.
And it worked brilliantly. Not only did it shrink the prostate index back down to normal levels, but it drastically reduced the chemical alarm bells in the tissue.
The pro-inflammatory cytokines, right, like IL-6, GCSF, and MCP1.
Yes. The specific chemical messengers that immune cells use to scream for backup and cause swelling were just silenced. The entire inflammatory cascade was broken.
Wow. So to recap this section, blocking JMJD3 with the drug GSKJ4 stops prostate growth in both chemical, the LPS, and hormonal, the testosterone animal models.
Exactly.
Which poses the ultimate question. Could a single pill, one targeting an epigenetic eraser, eventually replace the need for an aging man to undergo prostate surgery?
What's fascinating here is that we are looking at a fundamental paradigm shift in how we approach this disease entirely.
Right.
We aren't just blindly trying to relax the muscles around the urethra anymore. And we aren't just bluntly blocking all testosterone and forcing patients to deal with those side effects.
We're targeting the root epigenetic cause.
Exactly. BPH is no longer just a mechanical inevitable issue of getting older. It is a highly specific, complex cascade that we now actually have the blueprint to intercept. Targeting JMJD3 with drugs like GSKJ4 represents a massive leap forward in offering, as they say, the best treatment for the best patients.
Where does this leave the future of urology and patient care?
It basically brings us to the era of precision medicine. If a patient comes into the clinic with BPH that is driven heavily by this inflammatory cascade, giving them a targeted epigenetic inhibitor like GSKJ4 is a game changer. We are moving from mere symptom management to actual disease modification.
It's incredibly empowering. I mean, we took a notoriously murky aging disease and broke it down to its studs. Chronic inflammation acts as the spark. That spark produces the epigenetic eraser, JMJD3. The eraser runs into the nucleus, finds the TGF1 growth gene, and rips off the do not disturb sign. The growth gene turns on, the cells multiply, they refuse to die, and the prostate swells.
Right.
But now, by understanding that software, we know exactly how to block the eraser and rewrite the sticky notes.
It just highlights the sheer power of epigenetic research. I mean, we aren't entirely helpless to our DNA if we learn how the cell reads it.
Which leaves me with one final provocative thought for you to mull over today. We've just traced how an epigenetic eraser triggered by everyday inflammation, is the secret culprit behind benign prostate growth in aging men.
Right.
But inflammation happens everywhere as we age. It happens in our joints, in our brains, in our cardiovascular systems. So what other age-related inflammation-driven diseases in our bodies are secretly being controlled by these exact same removable sticky notes on our DNA?
That's a huge question.
Right. Are we looking at the underlying mechanism for arthritis? Or perhaps certain pathways in Alzheimer's? Could mastering epigenetics, learning exactly how to put the do not disturb signs back on our genes, be the ultimate key to controlling how we age?
And then that is the exact frontier researchers are stepping into right now.
The next time you see a donut, remember,
Mm-hmm.
it's not just about the dough swelling. It's about the microscopic sticky notes telling it to expand. Thank you for joining us on this deep dive. Stay curious.